Good afternoon, ladies and gentlemen. We're going to talk bacteria. This is a super long set of notes. The good news for you is that I still only get to talk 10 minutes because that's the time limit I have on this. So please make sure that you have your computers open and a tab open to your bacteria notes so that you can take notes while I'm talking, and then pause me whenever you need to have a little extra time to get your typing going. All right, let's talk bacteria. First of all, we talked in a previous unit, we said that bacteria are known as prokaryotes, and evolution says that we think bacteria are the oldest form of life on this planet. We think that life first showed up on this planet about 3.5 billion years old, a billion years ago, but the planet is about 4 to 4.5 billion years old. Scientists even believe that mitochondria and chloroplasts were actually once free-living bacteria that moved into larger cells, eukaryotic cells, for some kind of a protection, and they provided those cells with the services of making ATP or glucose and oxygen, et cetera, et cetera. It turns out that mitochondria and chloroplasts have their own DNA, and they divide separately from the rest of the cell. So that's kind of good evidence that perhaps they were free-living bacteria at one time. All right, the bacteria make up the most dominant life form on this planet, and there's so many bacteria that they've actually been divided into two domains. The eubacteria is what we're going to spend most of our time talking about. They're what people often refer to as germs, although very few of them make us sick. They're the ones that we tend to come in contact with. The archaebacteria, archae means ancient, they're an older form of bacteria, and they tend to be found in more extreme environments. I'll just mention a few archaebacteria, and then we'll spend the rest of the time talking about eubacteria. The first example of archaebacteria are the methanogens. We've talked before about how cow farts are causing global warming. It's the methane in cow farts. Well, that's coming from the bacteria that live in the cow guts. They're called methanogens. They're anaerobic, which means they survive without oxygen, and they release the chemical methane, which is a very potent global warming gas. Methanogens are also found in swamps and in sewage, and then in the intestinal tracts of animals. The next kind of archaebacteria are the extreme halophiles. Halo means salt, and you know phil means love, so they love salt. They're able to survive in extremely salty conditions, which would normally be hypertonic to most other organisms. So the extreme halophiles can live in places like the Great Salt Lake and the Dead Sea. And then the last kind of archaebacteria that I'll mention are ones you may have seen if you've ever traveled to Yellowstone National Park. They're called thermoacidophiles. Thermo refers to heat, acid is acid, and phil means they love it. So they like hot, acidic environments. They're found in the hot springs, in volcanic vents, and in cracks in the ocean floor. We don't come in contact with archaebacteria on a regular basis, so we're not going to spend a whole lot more time talking about them. All right, so eubacteria. You get infected with eubacteria. However, less than 1% of eubacteria are actually harmful to humans. The vast majority of bacteria couldn't care less about us, and only a tiny percentage actually make us sick. So many help us. They help us digest our food. They help us make vitamins in our intestines, and they help us fight off disease-causing bacteria. It's actually a good thing to have ordinary bacteria in your gut because they take up space from bad bacteria that could move in. Some bacteria are also used in making foods like yogurt and cheese. So what do eubacteria look like? Well, we've got some information. We know that they're prokaryotic, we know they're super small, and we know that they don't have a nucleus. They have an outside capsule that protects them and allows them to stick to surfaces. That's why soap is effective. Soap makes your skin slippery, and it allows the bacterial capsule to slip right off your hands. They have a cell wall. It is a different cell wall than plant cells. Their cell wall is made of a giant macromolecule called peptidoglycan. The cell wall of plants is called cellulose. They have DNA, but no nucleus. And then they have little pili that are hair-like structures that help bacteria attach to surfaces, and they help the bacteria to trade DNA. The pili are actually hollow, and DNA can travel through the pili to another bacteria, and we'll talk about that a little bit more later. So how do bacteria reproduce? Well, they basically reproduce through mitosis. Only for bacteria, we call it binary fission. It's a little bit different because they don't have a nucleus. But the basic idea is that they're cloning themselves. They simply split in half. So if all they do is clone themselves, KJ, how is it that bacteria evolve? Because we know they evolve. They've evolved resistance to antibiotics. Well, there's 3 ways that bacteria can evolve. They don't have sex, but they can exchange DNA with other bacteria. So the 3 ways. The first way is called transformation. Bacteria are able to suck up the DNA of another bacteria in the environment, another one that's dead. So it's like if a person died in front of you and you could just suck up their DNA and then suddenly have their traits. That's what bacteria can do, and it's a way that bacteria can evolve. Another way in which bacteria can evolve is through a process called conjugation. They can trade DNA through their pili. It's sort of like sex, but there's not male and female really in the bacteria, and it's definitely not meiosis or half of the chromosomes, that kind of thing. But it's called conjugation, and it's a way to get new DNA. The last way to get new DNA is called transduction. This one's a little bit less common, but sometimes, and you remember this from our virus notes, on the diagram, you see a virus that has attached onto a bacteria. It's injected its DNA. And as that bacteria is building new viruses, some of the bacteria's DNA gets trapped inside some of those new viruses. That's called a defective phage or a defective virus. If that virus then goes and infects another bacteria, it'll inject the first bacteria's DNA into the other bacteria. And that's a way that the new bacteria can get new DNA. It's called transduction. It's kind of a weird thing. All right, so KJ, we knew that from viruses made us sick by causing our cells to burst open, basically. So how is it that bacteria make us sick? Well, they make us sick by releasing poisons. And those poisons or toxins can kill tissue and cells. It can paralyze our cell machinery, and it can cause an overreaction of the immune system. In the end, bacteria reproduce so incredibly quickly. They can divide every 20 minutes. So think about the staggering numbers. They can actually crowd out healthy tissues and disrupt our normal functions. So how is it that we fight bacteria? Well, here's the good news. Remember, viruses, we don't really have a medicine for. We only have vaccines for viruses. For bacteria, we do have things called antibiotics, which are drugs that interfere with bacterial cell functions. In the diagram, what you see are 4 Petri dishes, and I'm going to let you read about it on your own instead of taking my talking time. I will definitely test you on what that picture is about, so you can read about that and answer a question. Think critically, see if you can answer this question. All right, so what exactly is antibiotic resistance? On that photograph that we just saw of the Petri dishes, sometimes the bacteria grew over the antibiotic. What's allowing that? Where are we getting antibiotic resistance from? How's that happening? Well, first of all, this is how it goes. A group of bacteria is exposed to an antibiotic. That means you get sick, and so you take some antibiotics. Susceptible bacteria are killed by the antibiotic. What that means is the weak bacteria die, and what's being left unsaid is the strong or resistant bacteria are surviving. They survive your antibiotic. The few remaining, step number 3, the few remaining mutant bacteria who are resistant to the antibiotic are usually killed by the immune system. So when you take an antibiotic, the weak ones are killed by the antibiotic, the mutant ones are killed by your immune system, and you get better. Problem is, people with weak immune systems, for example, the elderly, the very young, AIDS patients, people who've had organ transplants, they might not be able to fight off those last few strong bacteria. So now those strong or mutant bacteria can start multiplying within that person's body, and they produce an entire colony of resistant bacteria. Now a new antibiotic has to be used on that resistant bacteria, and the process can actually repeat itself. There are some bacteria now resistant to all known antibiotics. There's some versions of tuberculosis that we don't have a cure for anymore. And if I have a normal immune system and somebody coughs on me, one of those super immune bacteria, there's nothing I can do about it either, even though I have a normal immune system. So antibiotic resistance is a really big deal. So how can we prevent antibiotic resistance? Well, first of all, it's really important that we only take antibiotics when we have bacterial infections. Remember that the vast majority of colds are caused by viruses, the rhinovirus, so taking antibiotics doesn't do us any good. Misuse of antibiotics, like having antibiotics in our soaps, could actually be leading to more antibiotic resistance. The other thing we need to do is make sure that if we are taking antibiotics, we completely finish the antibiotic prescription, even if we're feeling better. I remember taking antibiotics about 10 years ago, and I felt better within 24 hours. I had a 10-day supply that I had to keep taking. If you take the entire prescription, you destroy all the susceptible bacteria, all the weak bacteria, and that leaves your immune system free to focus on the resistant or mutant bacteria. So it's really important that we take antibiotics carefully and that we are not careless with such a powerful medicine. All right, that's it. I managed to fit it in 10 minutes. You guys have a great day.